Okay, do you see quiz 4?
Yes.
Okay, I just quickly want to go over the answers so that you'll all know, in case you got any wrong, what the right answers are. So the first question was match the polymer with the monomer. Monomer is listed on the left. And they are, the answer is given here in this slide, which was in a lecture preceding the quiz. A nucleotide, the polymer is a nucleic acid. Amino acid monomers make peptides. Monosaccharides make polysaccharides, and fatty acids make complex lipids. Okay, so that's number one. Number two, indicate the substance that is not a type of lipid. So one of the slides you had gives all the different types, major classes of lipids. And the one item that isn't on this list, that is on this list, is spider silk. Spider silk is a protein, which was on another list that you had. Next was true or false. DNA and RNA molecules are constructed from nucleotides, each of which contains a phosphate group, a 5-carbon sugar, and a nitrogenous base. And that is true. One of the slides we had explicitly states that, and here's a diagram of it. Which of the following is not a difference between DNA and RNA? Well, it's not the first one, because that is a difference, double-stranded versus single-stranded. It's not the last one, hydrogen bonds versus no hydrogen bonds. It's not next to last, long versus short. And it's not thymine versus uracil. The one that doesn't fit is covalent bonds versus ionic bonds. And that's because the backbones of both DNA and RNA are held together by covalent bonds. And that is illustrated in this slide, which you had previously. Then true or false, all amino acids have 2 functional groups attached to a central carbon, one of which is a carboxyl group and the other is a phosphate group. And that is false. The reason is shown on this slide. All general structure of amino acid is amino group and not a phosphate, but rather a carboxyl. So the answer to that one is false. And then I showed you a drawing and said, indicate the arrow pointing to the peptide bond. And most of you got this right. It's B. Remember, the peptide bond is between the C on the carboxyl and the N on the amine. So it's C double bond, N, H. And that came from this slide. Then number 7, incomplete proteins are those missing one or more essential amino acids. The proteins of some plants are incomplete. For example, I have a list there, different plants missing different essential amino acids. So the question is, indicate 2 plants that would provide all essential amino acids. Most of you said rice and beans, and you can see why that is. Rice is missing lysine, beans have it. Beans are missing methionine and tryptophan, and rice has it. So, but any combination of those vegetables that provide all the essential amino acids was considered right.
Please. Beans and peas.
A question? Oh, okay. What is a gene? A gene is a section of DNA that codes for another molecule, usually a protein. So that is the answer to this question. Some of you had that explicitly, others had some variation of that. The sequence of nucleotides for each gene is unique. So there are no two genes that have the same nucleotide sequence. The nucleotides are read and interpreted in groups of 3 letters. It's not part of this question.
Is that true for twins as well?
Oh, which? Whether they're the
The
same?
genes are unique?
Yeah, if twins, they're not unique. In other words, each member of a set of twins has the same gene. Should have exactly the same sequence.
Mm.
Kind of the point that was being made is that, for example, hair color, if it's brown, that would be a particular sequence, but it would not be the same as the sequence for eye color or something along those lines. So individual genes have different sequences, but it's possible for someone to have the same sequence as someone else for a particular gene. So twins can have the same sequences because their genes are identical. But the question is saying in DNA, the genes for each different trait are individual to one another.
Yeah, good point. Okay, going to the next one. Isomers are 2 compounds. Hang on a minute. Isomers are 2 compounds with the same what, but different in what? So they're 2 compounds with the same chemical formula, but different structures. And we accepted a number of different variations on this, as long as it meant the same. For example, I remember some of you said different arrangements. Okay, they are different arrangements. And finally, the last one, a monounsaturated fatty acid differs from a saturated fatty acid by having a what? It's a single double bond in the chain. And that's from a slide that we had, which you can see here. It's a saturated carbon chain, except for at one point, there's a double bond. So that makes it a monounsaturated fatty acid. If there were 2 or more, it would be called a polyunsaturated fatty acid. And you'll often see that on food labels. The lipid in that particular food is polyunsaturated. It's supposed to be more digestible. Okay, any questions about quiz 4? All right, and as I said earlier, quiz 5 will be on Wednesday this week. So we'll take this down.
Dr. Yoder, do you have anybody waiting in your waiting room?
I don't see anyone.
Okay, I had an email from somebody, but it was like 3 minutes ago.
Oh, wait, no, I just got 3 that popped up. So I've admitted them. Okay. Now I'm going to share my screen with you. Okay, you should be seeing mutations and the CFTR protein. Everybody got? Oh, okay, good.
Yes.
Okay, what is the CFTR protein? Well, it's encoded by the CFTR gene. And just to review what we went over previously, where is the gene located? It has a location code. Does anybody remember how to read this code? 7Q3-2.1.
Zach Mushler. Oh, sorry, go ahead. Okay, I'll go. The location code is red chromosome, leg, I believe it's portion, band, and subband.
You're on the right track. What does the 7 indicate?
7th chromosome.
Yes. And the Q?
Long leg.
Yeah. And then-
Technically, they're arms.
Yes, thanks. Thanks, Scott. They're not legs, they're arms. And then 3, the number 3 is the region, 2 is the band, and 0.1 is the subband. So that tells you where on chromosome 7 FTR gene is located. Now it has, it's a very long gene, over 250,000 nucleotides. However, there are only 1,480 amino acids in the protein. So there are many more nucleotide bases than would code for the 1,480 amino acids. How to explain this? The answer is introns, primarily introns. And finally, the normal function of the protein is to transport chloride ions across the plasma membrane. So let's consider introns and how they affect the structure of a gene. Enough of coronavirus for now, and we'll get back to the... Where did that come from?
But teacher, the intron doesn't have the function here.
Yeah, introns, well, I can't say that they don't have a function. They don't have a coding function. Enough of coronavirus. They don't have a coding function.
Yeah.
So here you can see the relationship between exons and introns. The wide gray bars are exons, and this is the CFTR gene. The narrow gray bands are the introns. They don't code for proteins. So in the CFTR gene, there are 27 exons and 26 introns. There's always one more exon than introns. You can see why that is, that there has to be a start for every gene, and that's in an exon, and there has to be an end in every gene, and that is in an exon. So then what happens? More about, I have some voice in here that I forgot to delete, so I'll just have to delete it slide by slide as we go. So intron splicing in messenger RNA. On the left, you see a complete gene, the DNA, and then exon and intron alternation. That DNA is transcribed into pre-mRNA, and this occurs in the nucleus. Then the pre-mRNA is spliced. So the exons are taken out. The exons are spliced together to give the mature message, which is then translated to the primary structure of the protein, amino acid chain, and that eventually is cross-linked to form the tertiary structure. So the things to remember here are intron splicing occurs in the nucleus. It only occurs in the nucleus, and it only occurs in eukaryotes. Prokaryotes do not have introns. So you're probably asking, why are there introns? Well, some are ribozymes, which we'll talk about in a bit. Some are regulatory sequences, and others we don't know. So now we have an additional type of RNA. We've talked about mRNA, tRNA, rRNA, and now ribozymes, which are catalytic RNA. The name comes from ribonucleic acid enzyme, ribozyme. So what ribozymes can do is catalyze the hydrolysis of their own phosphodiester bonds. In other words, they can cleave a chain of either DNA or RNA by cutting at between the phosphate and the sugar. So you can see that illustrated here at the bottom. It's an RNA message. The ribozyme comes along, it recognizes a point, and it will cut there. And it cuts every message at the same place, which is shown here on the right. On this slide, sorry about that. I'll just have to keep doing that because I forgot to take them out. All right, how might a ribozyme regulate gene expression? Well, here's an example where there's a message and a ribozyme and a gene. If the ribozyme is inhibited, so it's non-functional, it can't cut the RNA, you get gene expression because the RNA stays intact. If the ribozyme is not inhibited, it cuts the RNA and there's no gene expression. So ribozymes can determine whether or not a gene is expressed. This shows one way that a ribosome regulates RNA. Introns have options. There's what's called alternative intron splicing. And this is yet another means to regulate gene expression. And it's a way of introducing variation into the genome, and it promotes evolution. So there can be full splicing. Here you have exons 1, 2, and 3 separated by introns. Well, if both the introns are taken out, then you get the full section 1, section 2, and section 3. Now there can be exon skipping. So if exon 2 is skipped, you'd end up with a sequence that's just 1 and 3. There can be alternative 5' slicing in which you get just part of exon 2. There can be alternative 3' splicing in which you get only the other end of exon 2. Or there can be intron retention. Some genes actually read the intron, in which you'd have exon, intron, exon, exon. So these are all alternatives which are primarily involved in intron splicing. Okay. Ribosomes are one means of the CFTR protein, located on the surface of many cells in the body. It's a single chain of amino acids. They're grouped into 5 functional regions called domains. 2 transmembrane domains, 2 cytoplasmic nucleotide binding domains, and 1 regulatory domain. So here you see a diagram.
Since the CFT
a diagram of the CFT protein through the membrane. So the background here is the phospholipid bilayer of the membrane. The protein goes all the way across it, from inside the cell interior, to the cell exterior. And there are these transmembrane domains, I think there are 10 of them, which form a circle, which makes a channel. The channel is for the export of chloride ions. So then you see that there are 2 nucleotide binding domains. So what are they? You see that here. And here's something you recognize: adenine. It's a base, it's a nucleotide. And the adenine base is can be a triphosphate, ATP. And you're familiar with ATP. It's the so-called energy currency of the cell. So ATP has a high energy bond here, the last bond in this phosphate. Now, if that bond is cleaved, the molecule goes to ADP, adenosine diphosphate, with the release of energy. Now, the transport of chloride ions is active. It's an energy requiring process. And where does that energy come from? Right here. The energy from ATP when it's converted to ADP. Okay, this shows the production of the normal CFT protein. So this includes all the steps from the encoding of the protein in the chromosome to the final destination of the protein in the plasma membrane. So the DNA is transcribed to RNA. Remember, it's the RNA that the DNA never leaves the nucleus. RNA can through these nuclear pores. And just outside the nucleus are what's called the endoplasmic reticulum. That's where the ribosomes are. So the ribosomes then translate the mRNA into protein. So now we have the globular protein, which goes to the Golgi apparatus. And the Golgi apparatus does a process called decorating the protein. And by that, it means attaching sugars. And once the sugars are attached, it's transferred to vesicles. And finally, the vesicles transfer that into the plasma membrane. You have the mature protein, and if it's wild type, it's functional. It transports chloride ions. And there's a video here that I'd encourage you to open and watch. I don't want to take time for it now, but please do it on your own. I've taken 3 screenshots from that video. It's a hand drawing explaining how the CFTR gene works with the introns and exons, the splicing. and the decorating with sugars of two types. There are core sugars and there are complex sugars. And this decorating goes on in the Golgi, shown here. Here's the nucleus, here's the ribosomes and the endoplasmic reticulum. And finally, the protein goes to the Golgi, where it's decorated with sugars, and that's what's shown here on the right. So what are the core sugars and the complex sugars? The core sugars are unbranched. They're just long chains of usually glucose molecules. Complex sugars are branched. So that's how to remember the difference in the decoration of proteins. The core sugars are single chain or are unbranched chains, and the complex sugars are branched.
In the video,
Okay, so we'll come back to CFTR later, but now I'd like to talk more specifically about mutations. I'm going to come back to cystic fibrosis. It is a genetic disease. The only way it happens is if there are mutations in the CFTR gene. But more generally, I want to deal with mutations. The definition, and this is something you should remember, it's any change in the nucleotide sequence of DNA. It may result in a change in the amino acid sequence or not. And if a mutation does cause a change, the protein may now function incorrectly. And you'll see that when we come back to CFTR. Mutations are the basis for permanent variation in organisms. There are other ways for variation to occur that are not permanent. And mutations can cause diseases in humans and in other organisms. And the example we're going to focus on is the disease cystic fibrosis. So what causes mutations? Well, there are a number of ways that they can occur. There can be copying errors. Remember, we went over DNA replication. There was 6 steps, 6 enzymatic activities to copy a section of DNA. That process is quite complex, and it can have errors in it. And if there is any error whatsoever, that's a mutation. There are chemicals that can damage DNA, so-called intercalating agents, which insert themselves into the nucleotide chain. And there are carcinogens. Mutations can be the result of radiation. I'll talk about an example of that next. But finally, I have emphasized here genome editing, because genome editing is the revolutionary process that's just been discovered recently, and it's completely changed the face of biology. And I'll tell you why a little later.
Excuse me, Professor.
Yeah?
So for genome editing, so it might be good, right?
Yeah, genome editing is good because it's controlled by humans.
Yes. Okay.
And I'll talk in some detail about, this is CRISPR, about how CRISPR is done and why it's such a powerful technique.
Yes, so mutation, they might be good or they cause disease. Sometimes they are good.
Yes, we always think of, or usually think of mutations as being bad. And very often they are bad. So for example,
Yeah.
if you get a mutation in your CFTR gene, that's bad. But if you want a mutation, then you try to make one. And genome editing is a very precise and efficient way of doing that.
Yes. Okay, thank you.
Okay, here's an example of how a mutation can occur as a result of, in this case, ultraviolet. In the DNA chain, there are bases called thymines, you know that, and they can be not necessarily next to each other, but close to each other. Now, if that DNA molecule is struck by UV light, it can cyclize and cause what is called a thymine dimer. Now, thymine dimers are formed, when they're formed, the bonds that hold them together are covalent. So they're very strong bonds. And just to the detail on that, remember the DNA has 4 nitrogenous bases. Thymine is just one of them. And these bases are called either pyrimidines, which are cytosine and thymine, or purines, which are adenine and guanine. And here to the right of that, you can see that pyrimidines always bond to purines and never to each other. So a pyrimidine thymine hydrogen bonds to purine adenine. And pyrimidine cytosine triple bonds, the 3 hydrogen bonds to purine guanine. Now, if that happens, you see on the right here, here are 2 thymines on the nucleotide chain within reach of each other. They're struck by UV light and a thymine dimer forms. Now, these red bars are covalent bonds between 2 thymines. And when that happens, these covalent bonds make a kink, a crimp in the DNA. So that now you remember when copying goes on, a helicase breaks these hydrogen bonds and you open up an open space for DNA polymerase to fit in. And when DNA polymerase 3 comes along copying this strand of DNA, it comes to this kink. And that kink could cause, in fact, almost always does cause, a change in the amino acid sequence. Either it puts in a different amino acid or takes one out, or it might even cause a deletion. So the pyrimidine dimer causes mutations by making a kink in the DNA. And that promotes copying errors by DNA polymerase. So now I've got a couple of examples here of what happens when a mutation occurs. So the mutation example is that the DNA sequence is given here, TAC, AGT, and so on. And then a mutation occurs so that this T is now copied in as an A. So what is the effect of that on the peptide? Well, you can figure that out by first transcribing the DNA to messenger RNA, and then translating that to a protein using a codon table. And this is homework for you. You need to go through this process, make sure you know how it works.
And this,
Now this is a second example of a mutation. So you have, again, the same original DNA strand, but now the mutation is at this G. It becomes an A. And again, what is the effect on the peptide? So you use the same process as the previous slide. You transcribe it to messenger RNA and then translate that to protein using a codon table.
This slide.
Now, what I've done here is summarize what happens with the normal sequence, the mutant 1 sequence, and the mutant 2 sequence. So let's just go over this. And then the normal wild type sequence, which is not mutated, here's the DNA, the RNA is translated, and you go to the codon table, and the peptide is met, ser, asparagine, cysteine, arginine, and stop. Now, in the case of mutant sequence 1, where this T becomes an A, so that the codon is now UCU instead of UCA. And if you go to the codon table, UCU, here's the first base is U, second base is C, third base is U. That's serine. And that's what you see here. So now the sequence goes met, ser, asparagine, cysteine. It hasn't changed. So this is a true mutation, which is silent. The peptide sequence does not change as a result of this mutation. Now, if we go to the third mutant sequence, where this G in the primary sequence is now mutated to A, and if you transcribe that into mRNA, the codon is now UUA instead of UCA. So if we look up UUA, first base is U, second base is U, and the third base is A, U. Yeah, UUA is leucine. And so now mutant 2 sequence leads to a change in the peptide chain. So instead of serine at the second position, it's leucine. So it's met, leu, asparagine, cysteine, arginine, and stop. And this is called a missense mutation. We'll talk about the different types of mutation in a minute. But the point here is that mutations can have various different effects, including no effect, a silent mutation, or a missense mutation, where the peptide is actually changed.
Now this is
okay, types of mutation. I've just talked about point mutation. A point mutation is one base pair change. So we have a table here that we're going to go over in a minute. And all the mutations in this table are point mutations, one base pair change. There are other types of mutations, and you need to know this. So mark this down as something that you need to know. What is a duplication? And most of these terms are quite descriptive. So a duplication, a piece of DNA gets copied and repeated. So you just have XYZ, XYZ, and it can be repeated over and over again. A deletion is just what it says. A piece of DNA is removed. An inversion, a piece of DNA is flipped over so that it reads backward. An insertion is a new piece of DNA is introduced. And a translocation, two pieces of DNA change places. Now it's possible to have a translocation where only one piece of DNA moves, but most translocation are so-called reciprocal translocations in that two pieces of DNA just change places with each other in the genome. And this can be quite a dramatic change. Now, the effects of point mutations, I'm referring now only to point mutations. A silent mutation has no effect. A missense mutation, we just had an example of that. One base pair alters the protein. A frame shift simply moves, takes the frame out of sequence. A nonsense mutation is a stop codon. So a single base pair change resulting in a stop codon so that the protein is never finished. This table on the right, table 14.1, just shows some examples of that. The first one, it's wild type, there are no mutations. The second one, this G changes to a C, and nothing changes in the peptide, so it's a silent mutation. In the next case, this second G here is changed to a C, and that substitutes proline instead of alanine. A nonsense mutation is a stop codon. And a frame shift, at the point of the frame shift, all amino acids downstream of that are different. So it dramatically changes the sequence of the protein. All right, so let's talk now specifically about the CFTR mutations. Over 2,500 different mutations are known in this one gene alone. So it's mutated many, many times. Most of the mutations are either point mutations, single base pair substitutions, or they're small deletions. The most common mutation is called F508del. Explain what that means in a minute. It's the deletion means that three base pairs have been deleted, and it leads to a loss of the amino acid phenylalanine. About 70% of the people, most of the people who have cystic fibrosis have this mutation. It's very common. But not all of the 2,500 mutations result in disease symptoms. Only about 400 of them do cause disease symptoms. And about 1 in every 31 Americans carries one mutated gene. One mutated gene does not cause cystic fibrosis. You have to have a mutated gene from each of your parents in order to have the disease.
Now that we've considered the
I just wanted to show you that the mutations can occur spatially quite different from each other. So we have the example here of deletion F508. It can be on homologous chromosomes. In other words, the very same mutation can occur on both homologs. Or deletion F508 can occur on one of the homologs, but on the other one, a different cystic fibrosis gene can occur. And that's called a heterozygote. And that can occur either on the other chromosome or on the same chromosome as the deletion F508 occurred.
I've included this slide.
So now let's talk about, I showed you the pathway of the CFTR protein from gene to plasma membrane. These various mutations, among the 400 odd mutations that have an effect, they can cause their effect at various different stages. So class 1 mutations, there's a mutation that doesn't allow transcription or translation into protein. So you get no protein at all, and no protein in the membrane. Or there can be mutations that change the amino acid sequence. There are mutations that change the decorating, and then mutations that change the transport of the membrane of the protein to the membrane. And finally, there are some mutations that cause the protein to become embedded in the plasma membrane, but they don't transport chloride ions properly. So there are 6 different classes of mutation.
You saw a picture, SIP.
And here they are, the 6 different classes of mutation. And this is a very busy slide, but if you look at it systematically, you'll see that it ranges from a normal CFTR protein through all 6 of the different types of mutation. So in the case of a normal protein, it goes through all the steps that we've just talked about, and you end up with a mature functional CFTR protein in the membrane. It transports chloride ions normally. Now, some mutations, and there are examples of them given here, they don't really make either RNA or protein at all. So the protein is absent from the membrane and no chloride ions can be transported at all. And there's another type 2, has the same phenotypic effect. Sometimes the protein is embedded in the membrane, but the channel is defective. And there are 3 different types of defective, 2 different types of defective channels. And type 5 is a functional protein in the membrane, but there are very few proteins, so very few channels, and chloride ion transport is reduced. And then finally, there are, decreased membrane stability. So again, it impacts the transport of chloride ions, and it's done inefficiently.
I have a question, Professor. It's Marissa.
Uh-huh.
Are you gonna want us to memorize, like, not memorize this chart for the test?
No, you don't need to memorize this chart, but you do need to know the pathway of the protein from gene to membrane and know what can go wrong in that pathway. That it can be at the level of the gene or the message, all the way out to the protein itself is in the membrane, but it's defective. Now, each of you, I've talked about the CFTR project. Each of you is going to be assigned a mutant. And you will end up at the end of the semester giving a report on your mutant, what's changed in your mutant. In other words, how the mutation has changed the DNA sequence, and what the effect of that, what is the functional effect of that, and given the function that is affected, what treatments are there for it? So those are the things that you will include in your report, and you'll be in teams, and each team will make a PowerPoint presentation about your mutation at the end of the semester. So that in a nutshell is why we're focusing on CFTR as an example of mutations, because you're going to talk about your own mutation later. I won't go over this now. This is for your purposes. It's simply a list that illustrates nomenclature of mutations. So if you just look at this first one, 254G to A, what that means is that at position 254, this is nucleotide position 254, the G is replaced with an A. So when you get the assignment of your mutation, it will have a code like that, which describes the mutation. I put this slide in, it's some summary points. I'm not going to go over these, but it's intended as a study guide for you to read these points and make sure you know the answers to the questions. Okay. And now I'd like to come back to CRISPR and the genome editing that has made such a huge impact on all fields of biology. First of all, the Nobel Prize for 2020 last year in chemistry was for CRISPR. And the two winners, the co-winners of the prize were this professor from UC Berkeley, Jennifer Doudna, and her counterpart, her cooperator in France called Emmanuel Carpentier. So they got the Nobel Prize for inventing CRISPR. So why is CRISPR so important? Well, sometimes you want to cause mutations. And so how do you do it? Well, one thing is you can look for replication copying errors, which I talked about. A pyrimidine dimer causes copying errors. Copying errors are relatively rare. And if you're relying on copying errors, you'll wait forever. You'll never get the mutation that you want. You can induce mutations in various ways. I just showed you how UV light can induce mutations, or ionizing radiation does the same thing. But these mutations occur all over the genome. So in principle, you mutate most or all of the genes in the genome, and then you have to sort through and find out to discover the one you're looking for. Chemicals that damage DNA are the same. They cause damage to DNA, but they're non-discriminating. They damage DNA all over the genome. So targeted mutations are what you want. And there are a number of different ways to make targeted mutations, but the champion of them all is CRISPR. It's called now genome editing.
What causes mutations? There are
So there are a lot of different reasons why you want mutations listed here. Why is CRISPR the method of choice? And this is what I want you to remember, is that CRISPR permits any mutation in any organism. So it doesn't matter what you're working with, a microbe, a human, a whale. you can cause a mutation in it, and it can be any mutation, any specific mutation that you want. So the 2 points are that CRISPR is site specific. You only mutate the target that you want. The second point is that it's universal. It works in all types of organisms across the biological spectrum. And this little picture here just shows forceps specifically taking a piece of DNA out of a DNA molecule. And so how does it work? What is the elegance of this technique? CRISPR stands for a cluster of DNA sequences that occurs naturally to help bacteria recognize virus RNA sequences. Cas9 is an enzyme that cuts foreign DNA in a specific place. To perform CRISPR, you need just 2 things. It's really, in principle, it's very simple. You need a guide RNA, which you can make in the lab. It's very short, usually 15 or 20 nucleotides. And that guide RNA targets the point in the genome that you want. So you need the guide and you need Cas9, the protein that cuts the DNA. So in the way that you proceed is to make your guide RNA and complex it with Cas9. So you have the Cas9 guide RNA complex. The guide RNA will go into the genome. So you take DNA from the organism of interest. If that's a human, you take human DNA. And you expose it to this complex of the guide RNA and Cas9. Now, the guide RNA finds its complement in the genome of your organism. So it matches up precisely, and this is what causes the specificity. It only matches up with the target that you've chosen and nothing else. Then, if this yellow is a spacer, the spacer tells Cas9 to cut the DNA at a specific point. And it only cuts at that point. Cas9 does not cut anywhere else in the genome, only where the spacer region tells it to. So now you have, you've cut the DNA. You can either put something in this space, in which case you'd have an insertion, or you can cut a piece out and have a deletion. So if you're
Excuse
just...
me, teacher, there is a lot of noise, background noise. I don't know.
What's that?
There's a lot of background noise. I don't know from which one.
Background noise?
Yeah.
Sounds like pots clanking.
Oh, Lourdes, could you... Okay, so I hope it's clear how CRISPR works. It's really very simple. You have a guide RNA and you have an enzyme that cuts where that guide RNA tells it to. And this is just an elaboration on what I've just told you, in case it's easier for you to understand in this way. I've got definitions of terms over here on the left. And if you go to the middle panel, you have the guide RNA and the spacer complexed with Cas9, the endonuclease. You mix them with your genomic DNA, and the guide will find your target, what's the gene that you want mutated. Cas9 cuts it, and then you can have, depending on what you want, you can have either an insertion or a deletion or a frame shift. So to summarize and to impress upon you the importance of this, CRISPR provides extreme target specificity in any organism. So it doesn't matter, as I said before, whether it's prokaryotes, eukaryotes, and among the eukaryotes, anything from primitive eukaryotes to very complex ones. So even viruses can be targeted. So you could imagine targeting coronavirus with CRISPR and destroying it. It can be used to correct genetic flaws. So for example, cystic fibrosis is a genetic mutation. If you replace the mutated sequence with wild type sequence, which is easy to do with CRISPR, you could cure cystic fibrosis. The big problem is you can do that in a cell. How do you translate that to an entire organism, to an entire human? There's plenty of opportunities for CRISPR in agriculture and in the environment, which I won't go into here because we're running out of time. So why should you be knowledgeable about CRISPR? It's because it's in the news almost every day. You may have noticed that in the past. If you haven't, you will now. And so when you read about CRISPR in the news, check it to see what is somebody doing with CRISPR now. It's revolutionizing biology, frequently in the news, and it's very likely that either you or someone you know will be affected by the use of CRISPR technology. And on the right here is all the different areas. Essentially, it's all of biology, all of medicine, and biotechnology. Laboratories in all these different areas are using CRISPR on an almost daily basis. Yeah, so that's the end of the slides. Next time, I want to start, I want to do a short bit on coronavirus and on coronavirus vaccines. I don't have time for that today, but that's the first thing we'll do on Wednesday. So I'm going to stop sharing now and ask if there's any questions or comments about today's session.
Zach here. We've been talking about the CFTR transport proteins, and you had mentioned that they use ATP to perform their function. I was just wondering the mechanism by which it uses the ATP.
I mean, how does the energy get transferred?
How does the energy get used? What is it used for? Because it just sort of seems to be a tube that stuff can flow through. So what is it used for specifically?
Yeah. Well, specifically, CFTR transports chloride ions. If there's no, and it's an active process. So if there's no energy available, nothing happens. Chloride ions don't move. If ATP is hydrolyzed to ADP, releasing energy, that energy is used to transport the chloride ion.
So is it like you're pushing against a voltage differential type deal, or?
Okay, you're getting into detail. Scott, do you have any more information about how the energy is transferred?
Sure. So CFTR is a gated channel. It literally changes its conformation from a closed to an open conformation or shape. In order to create that shape, it requires energy input, at which point the gate opens and allows chloride ions to move through. If there is not ATP there, then essentially the gate is closed and no chloride ions can move through. So how specifically in the kinetics of it, you don't need to really worry about that. That would
It was
not
more
for
for
your-
my own reference anyway, but okay, so that makes sense. The energy is used to change the shape of the protein itself.
correct, and thereby opening the gate and allowing chloride ions to move.
Gotcha. Thank you.
Okay, anything else? Next time I'll describe the nature of your CFTR project.
Professor, I have a question.
Uh-huh.
This is Marcus. It was about splicing and with the introns and exons. So the exons are the only ones that we care about.
Yes. Yeah.
But what happens to the introns?
They just get chopped up into monomers and recycled.
And then is there a certain thing that's doing the splicing, or is it just like a process that occurs?
No, it's done enzymatically. There are even little organelles called spliceosomes that in which there are the enzymes that do the splicing.
And remember that slide showed introns are not junk. There are admittedly many introns that we don't know exactly what they do, and there's a very active research into it, but introns are used as transcription factors or other regulatory gene expression factors. So just because it's spliced out doesn't mean it's necessarily destroyed. A lot of it may be, but they are working to figure out what additional effects these things have. They're finding out some pretty interesting things that introns do.
Cool. Thank you.
And for those of you that may have forgotten since the last time we mentioned it last class, I have a workshop at 2, so about 20 minutes from now, and be happy to help you out there. Anybody that's still working on the homework sheets, if you have questions, it's a good time to ask. If you have questions on this material, good time to go over it.